moving on. Number 2, we got Cipriani et al. Correlation between ontogenetic dietary shift and venom variation in Australian browns. That's Pseudonaja. Comparative Biochemistry and Physiology C, Toxicology and Pharmacology, 2017. So snake venoms, as we know, pretty rapidly evolving family of genes with going through gene duplication, functional diversification into families of venom genes. In Australia, some have evolved the prothrombin activating coagulopathic venoms. So activating the prothrombin clotting cascade in people's blood, or in mammalian blood, or prey blood, whatever kind of blood. This includes the Hoplocephalus, so the broad-headed and banded snakes, Notechis, the tigers, Oxyuranus, the taipans, Pseudonaja, browns, and Tropodecus, that's the rough scales. So an ontogenetic shift is a change throughout the animal's lifetime from one thing to another. So basically there's been some evidence of this before, I think from Timothy Jackson and down there at the Fry Lab, Jackson et al., 2016. They found that nocturnal foragers, things that are basically feeding on sleeping lizards at nighttime, are lacking in pro-coagulant toxins. So here they're kind of looking at the functional variation in the venom compared to the diet across the whole Pseudonaja genus and across some age classes. So just briefly, I think we've got to talk about blood clotting. So there's a bunch of clotting factors in your blood. This is just briefly for the audience. Imagine your a blood vessel. On the inside, you got your blood flowing through, and there's a bunch of coagulation factors in there that we're going to number from 1 to 7. Number 3 is actually on the outside of the blood vessel. So imagine you break that blood vessel and now the blood is flowing out through, and factor 3, which is on the outside, comes into contact with factor 7. It causes a bit of an enzymatic change, and now that complex of 7 and 3 can turn factor 5 into activated factor 5. Activated factor 5 can turn prothrombin into thrombin, and thrombin acts to transform in your blood a product called fibrinogen, which is soluble, to fibrin, which is insoluble, then precipitates out of the blood, which in combination with platelets forms a clot. Plasmin is another product in the which can be produced, which degrades these fibrin clots via fibrillysis, and there's other factors and cofactors involved. Got through that okay, I think. So look, they examined venom in 9 Pseudonaja and 3 juvie offspring using fluorometric enzyme activity studies, basically looking at the fluorescence difference between a substrate and a product to measure the enzyme activity. So just for example, you've got a tube of blood with some blood plasma, and you've got a fluorescent substrate for factor 5A. In this case, a protein called ANSN, which you add some venom. If Ep.6 is in there, it's going to bind to this substrate and fluoresce more. There's going to be more fluorescing ANSN. So high fluorescence indicates high Ep.6 activity, which itself turns prothrombin into thrombin. Thrombin turns fibrinogen into fibrin, which clots with platelets. All right. So,
so simple.
From the paper, once cleared by specific factors, the fluorescence group exhibits an increase in fluorescence. Specific factors were used to investigate different factors. Specific substrates were used to investigate different factors active in the venom or activated via the venom. So factor XA, plasmin, 7A, and thrombin, assessing the effect of venom on different components in the coagulation cascade, and then they mapped those coagulation activities to a phylogenetic tree from Skinner, 2005. And pretty cool, confirms the ontogenetic shift, which, pretty much to be expected. Non-coagulopathic juveniles turn to coagulopathic as adults in most of the Pseudonaja, except Modesta. Lower coagulopathic scores on its whole life, being mostly a lizard eater at night time. Is that correct?
Potentially.
Potentially?
Potentially. There's not a lot of agamids in their diet.
Yeah, okay.
So generally what you see with them is sort of eating
skinkers and
eating stuff at night.
Yeah.
You're tending to see agamids and skinks and stuff like that being highly present in the diet.
But not a lot of agamids.
Well, sleeping agamids.
Yeah. At night, there's a lot of agamids.
They're all asleep and they sleep on trees and shit like that.
Yeah,
so, or
right.
down in burrows. So, you get animals that aren't necessarily going up into trees, that are chasing agamids in trees and stuff like that. At the same time, though, we also don't know a hell of a lot about Pseudonaja modesta.
Yeah.
And I suppose I'm going to put a whole caveat here and say that Pseudonaja modesta, looking at the phylogeny of that, is actually more closely related to taipans than it is to brownsnake.
So you're thinking that it's going to be revised?
Well, the name was, there was a genus name that was erected for called Pseudonaja, which was by Wells.
Not a Pseudonaja?
Not a Pseudonaja. Yeah. Fucking original, isn't it? Wells
There's so many negatives in there.
Yeah. So it's a double
Not
negative.
a false cobra, yeah.
All right, fine.
Yeah, so...
Oh, there's a nerd joke for you. Oh,
There's just
my
like
God,
no
we're some horrible
backtracking
freaks.
that. Let's just add them on.
Yeah, well...
Just keep adding on to the name.
Yeah.
All right.
But, I mean, it was a name by Wells, so, you know, that'll be accepted any time this week. At the same time, though, I mean, there was a thesis study that looked at the phylogeny of Australian brown snakes, taipans, and black snakes, and now it's it's a thesis, so it's not technically published.
Okay.
So, but you're looking at that, and they suggest that there's going to be a rapid change in what would need to be the assessment of brown snakes and black snakes and a few other bits and pieces. So, I suppose the only concern that I've got with that is that if Pseudonaja modesta isn't actually a Pseudonaja, does that throw out the data?
Well, on that coagulopathic score, I mean, they're using Skinner et al.'s phylogenetic tree. So if that phylogenetic tree is wrong,
well, in Skinner's paper itself, in the conclusions of it, it does state quite clearly that
they're not sure about
Pseudonaja modesta is more closely related to taipans. So if it says that in the paper, which it does, right, then should they have shifted Pseudonaja modesta into Oxydomus?
In a different genus.
Or should they have erected a genus?
Not a Pseudonaja, yeah. Let's go. I'm all for not a Pseudonaja. I think it's fantastic.
It's a cool name, isn't it?
Yeah.
But,
Pseudonaja? Not a Pseudonaja.
So there's one other thing that I'm going to add to that as well, is that Modesta is actually a species complex.
Yeah, that's what I thought as well. There's more than one species there in the ringed browns. Is anybody looking at that?
Not now.
Not now. For me, the question was about geographic variation. So, like texties in eastern browns in Queensland are much more coagulopathic than they are in South Australia. The South Australia ones have a higher rate of taking out rat neurons with neurotoxin, and they're lower coagulopathic scores. So, how is that shift going to go? I mean, there's geographic variation on top of the ontogenetic shift. Yep. So that how that ontogenetic shift happens and at what stage, I wonder how that varies by geographic range myself.
I mean, there's rapid diversification in venom toxicity and stuff like that as well, depending on, even within the same species. I mean, so you've got age shift, and then you've got geographic shift as well, and then you've got poorly known phylogeography of some of those things as well. So,
and then you've got epigenetics on top of that could be doing God knows what.
Well, you use eastern brown snakes as an example. I mean, we regard the ones in Papua as textilis, and yet that paper that you were talking about in the last episode,
across the
shows
straits,
that the Papuan brown snakes are much more closely related to the isolated population of textilis that's found around Alice Springs.
Right.
So maybe some of these things might need to be looked at maybe a little bit more intensely, and go back and
do you think this is a species level changes or just locality?
I'm not a fucking geneticist. That's somebody else.
Okay, Yanni. Do you think this is a species level change, Yanni?
Yes.
All right.
You've heard it here first.
Don't listen to me. I got no idea what I'm talking about.
Yeah. It's on camera.
It's on camera now. I don't know. I think people are way too quick to jump on the fucking, let's call it a new species.
Yeah, absolutely.
But at the same time, too, I think at the same time, there's also a big problem where people are too resistant to change as well.
Yeah. I think I'm probably more on the resistant side. Not too resistant, but it does frustrate me when suddenly people just say, let's split this, split that, instead of just looking at the subtle differences within a species.
Well, look, I think one of the issues is that you get phylogeneticists that are going through and they look at the genes, and they're looking at the genes, and then you get a zoologist that's looking at the morphology or the ecology of a species or something like that. The two fuckers need to talk.
Yeah.
You know? And communicate. When they talk and they communicate, you get like a beautiful paper like Horner's Cryptoblepharus revision,
which was awesome,
fucking amazing,
and well done morphology, well done phylogenetics, absolutely jammed together really well.
Exactly, with confusing little fucking skinks.
All
right. At the same time, though, what comes out is people don't question effectively whether Paul was right on. He's gone through and he's done all the hard yards. It'd be much nicer if some of these newer papers that come out with taxonomy actually spent a bit longer and worked out. At the same time, too, we have a certain other problem with people coming in and using other people's data to slap names on things. And so there's a whole political side to taxonomy, which is really, really disappointing. So
I think
might have to talk about that another time. We might be here for 5 hours talking about that stuff.
I think we just need more people going back and refreshing data or double checking or getting more data rather than just constantly referring back to the one set that was put together.
Yeah, for sure.
One of the other things that was pretty cool in this paper, they predicted that rhinovorous animals would have the pro-coagulant venom, as opposed to things that are eating skinks. So things that are eating frogs at night time are perhaps coming across them in high metabolic rate because they're calling. Mm-hmm. So they're finding these calling frogs, which, as opposed to sleeping skinks, their metabolic rate is up. So you can hit them with the pro-coagulants and they will still manage to spread it around their body before it gums them up. Whereas those sleeping skinks, you don't want to mess with their circulation because you got to get that neurotoxin around to stop the heart.
Yeah.
So they figured, Pseudonaja guttata, little speckled brown, being a rhinophore eating frogs, just like red belly blacks, roughies and tigers, indeed does have those pro-coagulant venoms. So pretty cool. Just a nice little prediction that they pulled off there, I think.
Yeah. At the same time, though, with Pseudonaja guttata, they eat a shitload of mammals too.
Yeah, right.
Yeah.
So
I think
where the problem is that if you get somebody that's collecting Pseudonaja guttata, when they come to the surface out on the Mitchell Grass Plains, there's lots and lots of flooding. There's lots of flooding. There's lots of fucking frogs. If there's lots of frogs, what are going to be in the guts of the guitar?
Yeah, absolutely. Yeah.
Right. So I think they're
perhaps
eating a lot
rhinophore
more
versus
mice.
generalist might be a better term.
Could there be a shift?
Yeah.
A seasonal shift potentially in the
Seasonal ontogenetic
So
shift
when-
and
so when
prey type with venom.
Yeah. I don't know. More studies, more funding.
Yeah, there we go. Yeah, give these people some money.
Yeah.
All right. Look, I don't think we have time for this last paper, guys. I think we're going to have to leave it there.